to be talking today about autophagy and the relation with aging. As you know, aging is a multifactorial process in which conditions such as genetic impact, environmental, and metabolic conditions have an impact. Our interest is mostly on the metabolic component, and it emphasizes the fact that in order to produce energy, the cells generate also a lot of free radicals and byproducts that might result or might induce damage in the cell. So part of the theories of aging support that the inability to handle cellular damage might contribute to the process of aging. So how do these cells normally handle damage? Imagine that you have a protein and this protein is damaged by free radicals. The protein will get misfolded and it will expose these red regions that corresponds to the sticky or hydrophobic patches. To avoid this protein to stick around, the cells have different mechanisms of defense. First, they have the chaperones that are going to identify this area and are going to assist in protein folding. If protein folds, that resolves the problem. However, in many conditions, that's not sufficient, and the chaperones target this protein for destruction, and that occurs at the level of the proteolytic systems. This destruction is really a recycling process because the protein can be broken down in amino acids, and these amino acids can be utilized for synthesis of new proteins. These two components, the chaperones and the proteolytic systems, are in place and working in all the cells in the body as you are young. However, as you get old, the ability of the chaperones to recognize protein damage or the ability of the proteolytic systems to eliminate these damaged components seems to be compromised. That's going to result in the accumulation of these abnormal proteins that tend to interact with each other and form what is known as oligomer species of aggregates that are really toxic for the cell. So the emphasis of this lecture is trying to understand why the proteolytic systems, the activity of these systems, decrease with age, and what can we do to restore their normal functioning. There are different proteolytic systems in the cell. In one hand, you have the ubiquitin proteasome system, that is this very accurate and precise machinery that is going to take proteins and degrade them one by one. But in addition, you have a whole organelle, the lysosome, that is also completely devoted to degradation. The lysosome are single membrane vesicles that contain a large variety of hydrolases, proteases, lipases, glycosidases that are going to be able to degrade any component inside the cell. That includes soluble proteins, as in the case of the proteasome, but also proteins that are organized in the form of aggregates, as well as organelles. This is the way that your cell eliminates a damaged mitochondria or any other damaged structure inside the cell. And lastly, components that invade the cells, such as, for example, virus or bacteria, can also be degraded through this lysosomal compartment. And this degradation of cellular cytosolic components in lysosomes is what is known as autophagy, and that's the main topic of this lecture. So through this lecture, I would like to talk about what are the bases of the autophagy process, what are the main functions, and what are the main types of autophagy that cells perform, and then introduce the idea of the changes with autophagy with age and the consequences that this could have for age-related disorders. So going into the characteristics of the autophagy process. Lysosomes were described in the early 60s, but in the last 10 years there has been a lot of interest in this process, in the process of autophagy. The main reason has been a better molecular characterization of the different steps required for autophagy inside the cells. This is a result of screenings in yeast systems that have identified about 30 to 35 new genes that participate in this process. Having these genes have given us, in one hand, a way to really track this process in vivo, because now we have probes and different markers that can be followed in culture cells. But in addition, it has allowed us to modulate these genes, either eliminate them or overexpress them, and start understanding a little better what are the consequences of having too much or too little autophagy in your cells. This has generated a list of functions that has been now attributed to the normal functioning of autophagy. For example, has been shown that autophagy is required for cell differentiation, for tissue remodeling, in the mechanism of cellular defense, as well as in cellular death and in aging, as I will try to explain later. And also, the characteristics of the animal models with compromised autophagy have allowed to identify pathologies in which there is a primary defect in the autophagy process. Some of these diseases include cancer, neurodegenerative disorders, muscle diseases, and even some of the metabolic disorders very common in our societies, such as obesity or diabetes. So what are the main functions of the autophagy process? When autophagy was described, it was believed that the main function was to supply energy to the cells, because under conditions in which you don't have nutrients, your cells are going to activate autophagy to degrade part of the cytosol and obtain amino acids and free fatty acids that they will utilize to maintain the energetic balance in the cells. In addition, as I described before, autophagy can also eliminate damaged proteins and damaged organelles and contribute to the quality control of the proteome and all the components inside the cells. And lastly, because of this function in eliminating pathogens and toxic compounds that can be aggressors for the cell, it has also an important role in cellular defense. So when you have a cell in which autophagy is compromised, for example, as the cells get old or in different disorders, you're going to have, in one hand, cells that are unable to adapt to any kind of metabolic stress, such as starvation or, for example, high content of lipids in the diet. In addition, they are going to be more susceptible to the attack by different types of pathogens. And finally, they are going to have problems in the maintenance of their proteome, and they are going to start accumulating garbage, damaged products that cannot be eliminated from the cell. This is going to affect cellular function, and in many conditions might result in cellular death. So what are the different pathways or the different ways in which the cell can perform autophagy? As I mentioned, autophagy is just taking something from the cytosol and deliver it from to the lysosomes. So how do the cells do that? There are at least 3 different mechanisms that contribute to this delivery. One of these processes is known as macroautophagy, in which a whole portion of the cell, including, for example, mitochondria, ribosomes, and proteins, is going to be sequestered by this double membrane vesicle that seals, forming what is known as an autophagic vesicle or autophagosome. As you can see, this vesicle does not contain enzymes, but receive it after fusion with the lysosomes that is going to inject all these hydrolases that will lead to the complete destruction of any of the material sequestered in the vesicles. In addition, the cells can also perform microautophagy, in which a region of the cytosol is captured through invaginations in the surface of the lysosomes. In this case, you don't require the formation of autophagosomes, just small vesicles that pinch off from the membrane of the lysosomes into the lumen and get rapidly degraded. And lastly, in the case of mammalian cells, it is also possible to shuttle proteins from the cytosol into the lysosomes across the lysosomal membrane in a process that is known as chaperone-mediated autophagy. In recent years, most of the discovery has focused in macroautophagy and chaperone-mediated autophagy, and those are the ones that I will describe now in a little more of detail. So regarding the macroautophagy process, the characteristic or the morphological feature of this process is the presence of these autophagosomes. So if you look, for example, to this electron microscopy, you can see some dark structures that corresponds to mitochondria, some portions of endoplasmic reticulum, and then these big vesicles that contain inside parts of the cytosol that have undergone degradation. And you can actually track each of the steps of this process. You can see here, for example, a peroxisome that is going to be sequestered by this double membrane that is forming around the organelle. And in this case, you can see a mitochondria that is surrounded already by the double membrane vesicle, but you can still identify it as a mitochondria, so degradation has not occurred yet. And then, as the lysosomes fuse, you can see how the products or the material get degraded, and you end just with these lipids that eventually condensate to give secondary lysosomes. As I mentioned, through these screenings in yeast, it has been possible to identify about 35 genes that participate in each of the steps of this process. They work as complexes, and for example, you have an initiation complex that relocate to the area of formation of the autophagosomes, and then when the activation or regulatory complex also coincide in this area, this initiate the process of formation of the autophagosome. Through conjugation of proteins to proteins and proteins to lipids, this membrane is now going to elongate and eventually will seal, forming the autophagosome that fuse with the lysosomes for complete destruction. Thanks to the identification of these proteins, it is now possible to divide the types of autophagy in different subtypes depending on the cargo that is being degraded. You have still the in-bulk autophagy that corresponds to the degradation of a mix of components from the cytosol altogether, but you can also selectively degrade particular cellular components. For example, we talk about lipophagy, when you are degrading lipid droplets to obtain energy, mitophagy, when you are degrading mitochondria to eliminate any damaged mitochondria from inside the cell, and also many other organelles that gave rise to ribophagy, lysophagy, or aggregophagy. And the number of processes keeps growing as we learn more about this process. The other interesting feature is that because now we have all these different 35 genes, we also have their protein products, so we can utilize them to track this process in vivo. One of the markers that has been utilized in many studies is this LC3 protein that has been known to decorate the surface of the autophagosomes. If now we put a fluorescent protein attached to this LC3, we should be able to highlight the areas where autophagosomes are forming. For example, on the left, you can see a cell in which there is not autophagy, so LC3 shows as a diffuse green cytosolic pattern, but then as autophagosomes form on the right, you can see this panta that correspond to those vesicles that contain the material that is going to be degraded. So having this kind of tools allow us now to track the process of autophagy in real time in many different settings. The other pathway that we have also learned in recent years is this chaperone-mediated autophagy. This is a rather different mechanism in which proteins are targeted from the cytosol to the lysosomes one by one. All the proteins degraded by this pathway contain a pentapeptide, these 5 amino acids, that once exposed, it's going to be recognized by a cytosolic chaperone, the constitutive member of the HSP70 family. Once the chaperone and the protein gets together, they are targeted to the surface of the lysosomes, where they interact with this lysosomal associated membrane protein, type 2A or LAM2A, that acts as a docking station for this complex. Assisted by another chaperone located in the other side of the membrane, the blue protein, that is the one that you want to degrade, is going to undergo unfolding and it's going to cross the membrane and get rapidly degraded inside. Recent studies have identified about 10 different gene products that participate in this process. So what is the connection of autophagy and lysosomes with aging? For a long time, it has been, there are different questions that have been formulated in the aging community regarding autophagy. And the first one is, what are the changes that the autophagy pathway undergoes with age? So we knew for a long time that the lysosomes undergo major changes in all the cells as they age. There has very nice characterization showing a big expansion of the lysosomal compartment, an increase in the amount of enzymes delivered to these compartments, and in particular, the presence of undegraded material inside the lysosomes that has been described as lipofuncin. Lipofuncin is just the result of the cross-linking between undegraded materials and oxidative damage products that are going to produce this autofluorescent pigment that can be visualized in the aging tissues without any other kind of marker. So for example, in the picture on the right, you can see a section of the liver of an old rat, of 22 months rat, and even without any kind of staining, it's possible to see this granuli that corresponds to lipofuncin. And lipofuncin has been utilized for many years as a typical marker of aging. In addition of the alterations in the lysosomal compartment per se, it has been shown that the different autophagy pathways also decrease with age. Has been reports showing a decrease in macroautophagy and chaperone-mediated autophagy. In the case of microautophagy, the lack of markers to follow this process make that it's still questionable whether or not this process changes with age. So what we know about the changes in macroautophagy with age? If you remember the different steps that I highlight in the autophagy process, studies from different labs have shown that in different tissues as they age, there is an inability to upregulate autophagy in response to nutritional conditions or in response to different stimuli. This results in a poor formation of autophagic vesicles. However, it has been shown that the few autophagy vesicles that form remain in the cells longer because they cannot be properly eliminated by lysosomes, in part probably because of the accumulation of these lipofuncin or undegraded products inside the lysosomes. In the case of chaperone-mediated autophagy, we have also shown that the activity of this pathway decreases with age. For example, if you isolate lysosomes from young and old animals and try to recapitulate the translocation of proteins across their membrane, as you can see in the graphs, in the case of the old animals, shown here in orange, there is a marked reduction in the ability of these lysosomes to internalize these proteins. We engaged in a very systematic study to know what was the problem, and what we found is that the chaperones seems to be able to perform the normal function and targeting to the lysosomes till very late in life. And also the degradation inside the lysosome seems to be okay because chaperone-mediated autophagy only occurs in a subset of lysosomes inside the cell that happen to be different from those ones that accumulate the lipofuncin. A detailed analysis of the components in both sides of the membrane revealed that the main problem on chaperone-mediated autophagy in aging is a decrease in the levels of this receptor protein, this lysosomal associated membrane protein. As you can see here in the blood, these are lysosomes isolated from young and from old animals. These are two different sets. And as you can see here, the levels of LAMP2A decrease in the old group. And the same has been shown for other different species and even from cells of individuals of different ages. So this decrease in the levels of LAMP2A seems to be a common feature in all the aging organisms. So the other important question that we've been formulating in the field is what are the consequences of these changes in the autophagy system with age? So there has been many reports showing a clear connection between changes in macroautophagy and aging. And probably the most convincing, or at least the early ones, were those ones done in studies with C. elegans. It was shown that if in worms you block autophagy, this results in a shortening of the lifespan of these worms. Interestingly, as you know, there are different manipulations, either genetic or nutritional, that result in extension of lifespan in worms. And when you look at those animals, it is a common feature that all of them show an increase in macroautophagy. And actually, if you block macroautophagy in these long life species, they do not longer live so long, suggesting that you need this increase in autophagy in order to produce this longevity phenotype. Similar studies have been done in flies, and in this case, they show that just by overexpressing one of these autophagy genes, it seems to be enough to increase the lifespan of the flies. And actually, when you look at those flies, the process of neurodegeneration that usually associate with the aging of the flies was clearly slowed down. Finally, in mammals, one of the most convincing evidence of a connection between autophagy and aging came from the studies in different types of knockout animals for different autophagy genes. For example, I'm illustrating here the knockout of two different essential genes, specifically in neurons, and show how it is enough to reproduce a full display of neurodegeneration in these animals. And actually, if you look at the images of the different neurons at the bottom of the image, you can see the presence of these inclusions that corresponds to proteins that cannot be properly removed when autophagy is not working. So these studies support that continuous functioning of autophagy is required to maintain normal neuronal homeostasis and to prevent the accumulation of those aggregates. In fact, there are now reports for many other groups showing that when you have proteins that aggregate, they seem to be a target for degradation through macroautophagy, because if you remember, once proteins are aggregated, the other pathways can no longer identify them as substrates for degradation. So this has generated a lot of interest in trying to manipulate autophagy in conditions such as Alzheimer's or Parkinson to eliminate the pathogenic proteins that accumulate as aggregates in these diseases. The third question that we are interested in is trying to figure out how to fix the defect in the autophagy decline with age, and whether by repairing or restoring the normal function of these pathways we can slow down aging. So in the case of macroautophagy, something that has become evident is that there are many different ways in which the autophagy process can be altered, and this depends on the cell type and also on the cellular conditions. For example, there are conditions in which there is a defect on the initiation or formation of the autophagosomes, conditions in which you have a decrease in the levels of ATGs, and this has been shown to occur in some tissues as they age, but not only the effectors, also an increase in the inhibitors of these pathways, such as, for example, insulin signaling or mTOR signaling, will also have a negative impact in the formation of the autophagosomes. In these conditions, any intervention aimed to prevent the blockage of this pathway or to enhance the formation of autophagosomes should result beneficial. And for example, rapamycin, an agent that has been already shown to increase lifespan, even in rodents, will be able to block the inhibitory effect of mTOR over the autophagy pathway. In addition, in other conditions, the problem might be the identification of the cargo. For example, some aggregates cannot be degraded because they cannot be identified by the system, or damaged mitochondria might elude the surveillance systems of this autophagic process. Sometimes the problem is the presence of pathogenic proteins that might interfere with the autophagy machinery itself and prevent the recognition of the cargo that needs to be degraded. Moving along the autophagy pathway, there has been examples in which the problem is not in the formation of sequestration of the cargo, but in the ability of these vesicles to target or to fuse with the lysosomes. These include conditions in which the cytoskeleton is altered, or pathogenic proteins that might interact with the surface of these autophagosomes and lysosomes and prevent their interaction. And lastly, since the lysosomal system is the final compartment where all these materials are being delivered, any condition in which the lysosomal biology is altered will also result in alterations in the autophagy process. And that includes, for example, a deficiency in particular enzymes, inability to maintain the acidic pH of the lysosomes, or inability to preserve the normal stability of the lysosomal membrane. When we encounter these kind of problems late in the pathway, upregulation of the autophagy process or a regulation of formation of autophagosomes might not be longer beneficial, because you might end having all these compartments that don't have a way to be eliminated. In this case, it will be much more beneficial to add directly over the cytoskeleton machinery, if that's the problem, or try to eliminate these pathogenic proteins that interact with the autophagosomes, or ultimately try to resolve what is the lysosomal problem, either restore normal pH or provide the enzymes that are going to facilitate the complete destruction of the material that is no longer degraded as you get old. In the case of chaperone-mediated autophagy, we have also identified, as I mentioned before, that the problem seems to be in a decrease in the levels of this receptor protein, or LAMP2A. Since the complexity of these systems is less, we decided a while ago trying to restore the normal levels of LAMP2A and see if this was enough to recover normal activity of this pathway. So we decided to do this first in cells, using different senescent cells in culture, and trying to see if when we put back LAMP2A, this was enough to restore normal proteolytic activity. So for example, if we compare chaperone-mediated activity in the graph on the right, in young and old cells, as you can see here, you have a dramatic decrease in the degradation through this pathway in the senescent cells. However, if now we transfect them with the gene that encodes for this LAMP2A, this receptor of the lysosomal membrane, as you can see here, there is a restoration of proteolytic activity that gets very close to that we have in the early division cells, in the non-senescent cells. So these results encourage us to try to reproduce this restoration in vivo and see if by repairing or restoring normal chaperone-mediated activity, we could slow down any of the manifestations of aging. So to that purpose, we devised a transgenic animal in which we have included an extra copy of the LAMP2A gene. But we put it under the regulation of a tetracycline system, so we only activate this copy when we want. And the idea of doing that is that, because as you can see here, if we plot changes in chaperone-mediated autophagy with time or with age, as you can see here, it's not something that occurs since the moment you are born. It's normally when the animals reach middle age is when the activity of this pathway declines. So our idea was to activate this gene, this extra copy of the gene, in middle age, and basically maintain the same levels of this receptor as the animals when they were young. So then we compare how were the animals in which we have restored this pathway with the animals in which we haven't done any type of intervention. And the first thing that we looked, it was at the level of oxidized proteins, because we have previously shown that chaperone-mediated activity is required for the elimination of oxidatively damaged proteins. So if you look here, this is a bidimensional gel of the cytosol of a young animal, and we have used a probe that allows us to identify oxidized proteins. So each dot spot corresponds to an oxidized protein. If we compare young animals of 6 months old with old animals 22 months old, that will be equivalent to an 80-year-old person. As you can see in the old animals, there is this massive accumulation of these oxidized proteins that also move in the gel toward the right. So the question was, how are our animals? If we are able to maintain normal CMA activity, do they have similar levels of damage, or are they a little better? So what we found, as you can see in the plot on the right, is that the levels of oxidized protein were almost indistinguishable from those ones in the young animals, suggesting that restoration of chaperone-mediated activity was enough to prevent the accumulation of all those damaged products. We have now performed a complete characterization of these animals, and we have found that maintaining normal CMA activity till late in life is enough to preserve normal cellular homeostasis. So in addition of this decrease in oxidized proteins, we also have a reduced number of aggregates that you can identify here as these green spots staining for ubiquitin. We also have lower levels of lipofuxin. If you remember, I showed that the accumulation of these undigested products on the left corresponds to the inability of lysosomes to degrade materials, but when you look at these CMA restored animals, you do not longer see the presence of this lipofuxin material. And then also when we start looking, we found that mitochondria, endoplasmic reticulum, and organelles in general were better. And this was not only attributed to chaperone-mediated autophagy, but we found that macroautophagy activity and even the ubiquitin proteasome activity were also better preserved, suggesting that there is a crosstalk among these pathways, and that when one of them fails, the others take the load. So if we maintain or we prevent the failure on CMA, we prevent the overloading of the other systems. In addition, we also studied how were these animals responding to stress, because autophagy, as I mentioned, is part of the cellular response to stress. So when we compare the level of damage that you have normally in all animals, when we inflict damage by, for example, in this case, a hepatotoxic compound, and you look at the number of dead cells that are shown here as these bright spots, the amount of cells that die in the case of the animals that have better preserved chaperone-mediated autophagy is clearly less, and these are animals from the same old age. But then, of course, I'm showing here a liver that is cleaner and also responds to stress better. But the whole ultimate goal in aging is always to restore normal function. So we also perform functional studies trying to analyze how was the ability of the livers of these animals to eliminate toxic compounds. And as you can see here, in normal animals, in young animals, when you inject this particular compound, you need a time of clearance of about 1.5 hours. With the same amount of compound, the old animals need almost twice the time. But when you compare the animals in which we have restored normal chaperone-mediated activity, they have times of clearance very similar to the one in the young animals. And this occurs not only when we prevent the decline from the very beginnings on the activity of this pathway, but even if we wait for 18 months till the activity of the pathway is already down, and now we activate chaperone-mediated autophagy, we still have an improving function, suggesting that even late in life interventions might have a beneficial effect. So just to summarize what I want to cover during this lecture, as I mentioned, one of the main functions of the autophagy system is on the clearance of damaged proteins and altered organelles, so it's part of the defense against protein toxicity, and it seems to be very efficient in doing that in all types of cells. However, as I mentioned, with age, there is a decline in the activity of the different autophagy pathways, and part of the goals of the current research is trying to identify where is the defect and trying to restore or to repair this defect in order to maintain normal autophagy activity. As I mentioned, these pathways don't function in an isolated manner, but they crosstalk, so impairment in one pathway are normally compensated by another one, and on the same token, when you improve the activity of one pathway, it's going to have beneficial effects on the other. And just to end with a positive note, I think with the studies that we show in the case of restoration of chaperone-mediated autophagy, what it suggests is that a small improvement in quality control seems to have an exponential effect in cellular homeostasis, in the cellular defense to stress, as well as in cellular function, suggesting that interventions aiming at modulating these lysosomal pathways can have a great potential in the prevention, or at least in the slowdown of the aging process.